Okay, and we're back for another video here. So we've started to look at the molecular explanation for long-term potentiation, focusing on the role that the NMDA receptor plays in starting out this whole process of strengthening synaptic connections. And we've already said that an NMDA receptor is a special receptor because unlike most receptors in neurons, it's not just a neurotransmitter-dependent receptor. Most receptors just require the appropriate neurotransmitter to bind with them, and then they'll open up and allow their ions to come flooding inside. NMDA is unique in that it's a neurotransmitter and a voltage-dependent channel, meaning that two things have to happen for NMDA channels to open up and allow calcium to come flooding inside the cell. Those two things being the postsynaptic neuron has to be stimulated, which basically means that the postsynaptic neuron has to generate an action potential, and glutamate has to bind with the NMDA receptor. Those two things allow for calcium to come flooding inside the cell. Okay, so let's talk a little bit about how this process contributes to the strengthening of a synapse. I'm going to skip past this here for a little bit. Not too important, just sort of going into more detail on things we've already said. So what causes this strengthening of synapses? Why is the sort of influx of calcium important for strengthening a synapse between a presynaptic cell and a postsynaptic cell? So now we're going to start talking about the second of the two receptors that I said we were going to talk about today. So NMDA is important in sort of starting this whole process of long-term potentiation. And what actually contributes to the strengthening of a synapse between two neurons involves a second receptor called AMPA. AMPA receptors are going to be what's really primarily responsible for the strengthening of a synapse. And what's happening is that as a result of, and we'll look at this in more detail coming up, as a result of the molecular processes that we've already looked at involving the NMDA receptor, which when opened allows calcium to come into this cell, this is going to cause chemical reaction that leads to the insertion of more AMPA receptors into the postsynaptic membrane of the dendritic spine. So as we'll see, we'll look at a picture, I think on the next slide, that's actually sort of showing this process, where basically the reason a synapse becomes strengthened is that new receptors get added to the postsynaptic membrane. And AMPA receptors are again a glutamate receptor, which means that they are they bind with molecules of glutamate being the neurotransmitter. But AMPA receptors, unlike NMDA receptors, are more along the lines of sort of typical receptors, in the sense that they're only neurotransmitter dependent receptors. So these will open up when they bind with their appropriate neurotransmitter, which is glutamate. And so just like NMDA receptors, which control a calcium channel, so when NMDA was opened, calcium could come flooding inside the cell, AMPA receptors are sodium channels, or they control a sodium channel, I should say. And so what does that mean? That means that when an AMPA receptor has been stimulated, when it binds with a molecule of glutamate, that's going to cause this sodium channel to open up, which is going to allow molecules of sodium ions to come flooding inside the cell. And so one of the things that we know about sodium is abbreviated Na+. Sodium is an ion that is important for causing action potentials. So if you remember when we talked about our action potential earlier on, we said that what is an action potential? An action potential is a very brief change, let's see if I can draw it here, in the electrical charge inside of the cell, inside of a neuron. So here, T is time, and then this is voltage, measured in millivolts. So if you remember, we said that when a cell is at rest, the charge inside the cell, or more specifically the voltage across the cell's membrane, is negative. It's more negative inside the cell than outside the cell. And when a cell gets stimulated, what happens? It becomes more positive. It becomes more positive very quickly. What's happening to drive the, put a little 0 here because it actually goes past 0. It becomes more positive inside the cell for a brief period of time. What happens, what's happening inside the cell that leads to the charge becoming more positive, or the voltage across the cell's membrane becoming more positive? What's happening is that when a cell gets stimulated, what's driving this action potential, what's driving this depolarization, is sodium ions are coming flooding inside the cell. So if sodium is flooding inside the cell, it's a lot of positively charged ions coming inside the cell. That's causing a change in the electrical charge inside of the cell. It's causing, if you add a lot of positive stuff to the intracellular fluid of a neuron that starts out negative, if you add a lot of positive stuff, what's going to happen is the charge inside the cell is going to get more positive as you add more positive stuff to it, right? So sodium is crucial, is a really important ion that's involved in the action potential that causes that depolarization process, that first stage of the action potential, right? So what's happening if you add more AMPA receptors into the synapse, basically, you're providing more doorways for sodium to enter into the postsynaptic cell. So with more AMPA receptors present, it's easier for a lot of sodium ions to enter into that postsynaptic cell, which means that it will be easier for that postsynaptic neuron to fire. So it will be more likely that the postsynaptic membrane will fire, that it will generate an action potential, because it will be easier for a lot of sodium ions to come rushing inside of the cell. And that is, when that happens, if we're talking about it being easier for a neuron to generate an action potential, then what we're really saying is that the synapse has been strengthened, right? That the postsynaptic neuron is now more likely than it was before. It's now more likely to generate an action potential. So this picture here kind of shows the movement of AMPA receptors from an area called the dendritic shaft, which is down here, where these AMPA receptors start. So here again is a dendritic spine. So this is just a sort of an outgrowth from the dendrite of a postsynaptic neuron. again, there would be an axon terminal button here that would release neurotransmitters. Since we've only talked about one neurotransmitter, and that's really all we need to talk about, which is glutamate, right? So here is, in sort of I don't know what color you would call that, greenish, bluish color, is an NMDA receptor. So what did we say already about an NMDA receptor? This is sort of what starts the whole process of long-term potentiation. So as you remember, we said that two things have to happen for an NMDA receptor to open up. We said that, well, for one thing, an NMDA receptor controls a calcium channel. So it allows calcium, Ca2 here, to come flooding inside of the cell. But two things have to happen in order for the NMDA receptor to open up. The postsynaptic membrane, which is, all of this, needs to be depolarized, which basically means that the postsynaptic neuron needs to generate an action potential. That causes the magnesium that was serving as sort of like a roadblock to be ejected from the NMDA receptor, and so that now when a molecule of glutamate binds with the NMDA receptor, all that calcium can go flooding inside the cell. And so that's important because this is what initiates the process of moving AMPA receptors into the synapse. The entry of these calcium ions activate other ions, which initiate long-term potentiation, but through this process of triggering the movement of these AMPA receptors, which are starting out in the dendritic shaft, as a result of the influx of all of this calcium, these AMPA receptors will be triggered to move, to migrate from out here to the synapse, right? So you're adding all of these new AMPA receptors into the synapse. And what did we say about AMPA receptors? They're glutamate-dependent receptors. So glutamate will bind with them, as is sort of shown up here. And they control a sodium channel, right? So that when a molecule of glutamate binds with an AMPA receptor, well, for one thing, there are now many more, as we get down here, many more AMPA receptors after this process. So there are now many more doorways through which sodium can enter the cell, and a lot more sodium can get into the cell. And so all of this is contributing to making it easier for the postsynaptic membrane to generate an action potential. And if the postsynaptic membrane is more likely to generate an action potential, it means that the synapse has been strengthened, and the strengthening of the synapse is long-term potentiation.